Hi, my name is Ann Teal, and I will be a senior in the Chemical Engineering Undergraduate program here at the University of Michigan, Ann Arbor this fall. This summer, I had the opportunity to work in Dr. Nina Lynn's research lab here at the College of Engineering under the guidance of graduate student Michael Nelson. Today, I will be presenting about my summer research experience on the characterization of recycle stream in algal biomass processing. Currently, biofuels such as ethanol and biodiesel are commercially available. These first-generation biofuels are derived from plants. Primarily corn and soybean crops are used as feedstock for these biofuels. These crops are energy-intensive to grow, and recent studies show that there is not enough land area in the world to produce feedstock for substantial amounts of biofuel. Researchers are now turning to other sources of biomass. In other words, material that is derived from a living source. Algae is one viable option. First-generation biofuels require pre-processing to break down the crop's tough plant cell walls, whereas algae have no such rigid structure. Algae can be grown and harvested in either fresh or salt water, eliminating the need for vast areas of farmland. Researchers in the Savage Lab here at the University of Michigan have proven that after a high pressure and high temperature reaction of algae feedstock, bio-oil is formed. Bio-oil is a viscous organic material chemically similar to petroleum. One drawback is that much of the algae feedstock is not converted to bio-oil. Byproducts, including both an aqueous phase and solids, are also formed. These streams have the potential to be recycled into the feedstock or go through further processing to form more biofuel. The Lynn Lab is engineering microbes such as bacteria that could process these byproducts. To aid in this design process, we have been working on identifying and quantifying components of the aqueous phase. We performed kinetic growth tests in order to evaluate the bacteria's ability to grow in media containing glycerol, a component of the aqueous phase. These maximum growth rates compare different strains of non-pathogenic E. coli that have been engineered to quickly process glycerol. This summer, I worked mainly on the identification and quantification of the nitrogen and carbon compounds in the aqueous phase using analytical chemistry techniques, including high-performance liquid chromatography, or HPLC for short. HPLC is a way of analyzing an unknown solution by passing it through a tube of porous material. Different chemical compounds will bind to the tube, then release at different times depending on their chemical properties. As compounds leave the tube, detectors use properties of the chemicals such as ultraviolet absorption and refractive index to identify and quantify them. Using this technique, we have determined the concentrations of compounds such as glycerol and citric acid that are present in this aqueous phase. Knowing the composition of this byproduct will enable the rational design of a microorganism that can metabolize all of its components. This could lead to an overall algae fuel process that recycles most of its required nutrients with limited waste. Thank you for your attention.